Usually, when we think about treating a disease, we think about flooding the entire body with a chemical drug and just crossing our fingers that the collateral damage isn't too bad.
Right, the classic carpet bombing approach.
Exactly. We carpet bomb the system. But what if you could send a microscopic assassin directly into your cells? I mean, an assassin armed with a highly specific hit list designed to shut down disease-causing genes right at the source, long before they even have a chance to wreak havoc on your body.
It sounds entirely like science fiction, honestly. Yet it is the day-to-day reality of a field that is completely rewriting the rules of modern medicine. Today, we are exploring a remarkably powerful biological tool called shRNA.
Right, short hairpin RNA.
Exactly.
And I am looking at a massive stack of research notes in front of me today, and I have to admit, it is dense.
It is incredibly dense.
Yeah, we have studies spanning complex biophysics, cutting-edge clinical trials, bioengineering breakthroughs, and some really intense cellular biology. So, okay, let's unpack this. Our mission for this deep dive is to take all of this incredibly dense source material and translate it into something that actually makes sense for you listening at home.
Because whether you are just insanely curious about the future of medicine, or maybe you're a biotech investor trying to understand where the next massive leap forward is coming from, this is the frontier.
We are going to break this down.
Yeah.
But to really get what shRNA is doing, we have to start with RNA itself. If you remember high school biology, RNA is usually painted as just the messenger. Right.
They're just a courier.
Right. It carries the blueprint from your DNA in the nucleus out to the protein factories in the cell.
That is the traditional view, definitely. The classic central dogma of biology, DNA makes RNA and RNA makes protein. But what researchers have discovered over the last few decades is that RNA is not just some passive courier carrying a sticky note.
It does more than just deliver the mail.
Oh, absolutely. When RNA is structured in a very specific physical way, specifically folded into a short hairpin shape, it transforms from a simple messenger into an aggressive, targeted regulator.
It becomes a weapon.
It does. It becomes an active agent that can physically intercept other RNA messages and destroy them, effectively silencing the gene.
So before we can truly appreciate how this microscopic assassin actually cures a disease, I want to understand how it's manufactured right inside the patient's cell. The research describes this as an entire microscopic assembly line.
That's a great way to visualize it.
And I'm looking at the first step here. It says the creation of an endogenously expressed shRNA starts deep inside the cell's nucleus using a DNA template.
Right, so think of it as installing a new manufacturing plant inside the nucleus. We deliver a DNA template, a vector, into the nucleus of the patient's cell.
Okay.
That DNA acts as the master blueprint. From this template, the cell's own internal machinery begins transcribing or reading the DNA to build the shRNA. But the cell doesn't just read it automatically.
It needs a push.
Exactly. This transcription process has to be jump-started and driven by specific molecular engines called promoters. In the literature, you will see researchers using RNA polymerase successor or THR promoters. The most common ones are referred to as U6, H1, or 7SK promoters.
I actually have a note here about those polymerase, the third promoters, specifically U6 and 7SK. It says they are incredibly strict. Like, they have very rigid rules about what they will and won't transcribe. Why does that matter so much?
Well, it matters because of the underlying biophysics of how these enzymes physically grip the DNA. Pol-3 promoters have very defined initiation and termination sequences. Let's look at termination first.
Okay, stopping the train.
Right. If the Pol-3 machinery is chugging along the DNA track, building the RNA strand, and it encounters a run of just 4 thymidines, 4 T's in the genetic code, it acts as an absolute hard stop sign.
Wait, what? 4 T's? I mean, why not 3 or 5? What's so special about that specific sequence?
It all comes down to the physical stability of the molecules. When the enzyme is copying DNA into RNA, there is a brief moment where the newly formed RNA strand is temporarily bonded to the DNA template.
Like Velcro.
Sort of like a very weak Velcro. An RNA-DNA hybrid consisting of a string of uracils bonded to adenines, which is what you get when you transcribe a string of T's, is thermodynamically very weak. The hydrogen bonds holding them together are flimsy. When you hit 4 in a row, the connection is so weak that the entire complex literally falls apart. The machinery detaches, and transcription immediately terminates.
That is fascinating. So it's not a conscious decision by the cell. It's just pure physics. The track literally crumbles underneath the train.
Precisely. And it works on the starting end, too. Promoters like U6 have a strong thermodynamic preference for how they begin. They work best when the very first nucleotide of the target sequence is a guanine.
So you have to start with a G and avoid 4 T's.
Right. Designing these genetic constructs requires absolute, strict adherence to these rules. If a bioengineer makes a mistake and puts 4 Ts in the middle of their active therapeutic sequence, the drug will fail completely.
Because the factory just shuts down halfway through.
Exactly. The machinery will detach halfway through, and you get a broken, useless, premature piece of RNA.
Okay, so let's assume the bioengineer gets the code perfect.
Yeah.
The promoter does its job, the train stays on the track, and what rolls off the assembly line is this primary transcript.
Yeah.
The notes call it the pre-hRNA.
Correct.
And because of how the code is written, it automatically folds back on itself into a shape that looks like a hairpin, like a little stem with a loop at the top. But it says here it's not ready to be an assassin yet. It has to be processed by a microprocessor complex called DROSHA and DGCR8. What actually is a microprocessor complex? Is it just a single protein?
No, it's not. A microprocessor complex is a large cluster of different proteins working together as a single biological machine. In this case, it resides inside the nucleus. The DROSHA is an RNA's third enzyme. You can think of it as a highly specialized pair of molecular scissors.
Okay, scissors.
And its partner, DGCR8, acts like the hands that hold the RNA in place.
Because scissors can't cut paper if nothing's holding it.
Exactly. DGCR8 grabs the base of the pre-RNA hairpin, anchoring it, so that DROSHA can make a very specific, precise cut. This snip liberates a smaller, cleaner hairpin called a precursor shRNA, or pre-shRNA.
And there's a crucial detail highlighted in the research here. It says when DROSHA makes that cut, it leaves a very distinct signature, a 2-nucleotide overhang on the 3' end of the RNA strand. It's almost like a microscopic barcode or a tiny little handle sticking out.
That little handle is an absolute structural necessity for everything that follows, because now this pre-shRNA has to leave the nucleus.
Right, the factory floor.
Well, the nucleus is like the heavily guarded vault of the cell. The RNA needs to get out into the main floor of the cell, the cytoplasm, because that is where the target messenger RNAs, the blueprints for the disease, actually live.
So it has to break out of the vault.
Yes. To get out, it has to pass through the nuclear pore, and the nuclear pore is guarded by transport proteins.
The notes mention a transport protein called exportin 5. So if the cell is a factory, exportin 5 is the bouncer checking IDs at the factory door, making sure only appropriately processed materials get out onto the main floor.
I really like that analogy. Exportin 5 doesn't just grab any random piece of RNA. It physically feels for that specific structure, the hairpin stem, and that exact 2-nucleotide overhang left by DROSHA.
The handle.
Right. If the overhang isn't there, exportin 5 won't bind to it, and the hRNA is trapped in the nucleus. But if the ID checks out, exportin 5 encapsulates the hairpin and shuttles it through the nuclear pore into the cytoplasm.
Okay, the bouncer lets it out. It's on the main floor. Now it meets another enzyme called dicer1. If exportin 5 is the bouncer, dicer1 sounds like the quality control inspector. I'm reading that dicer1 physically measures the hairpin and then chops off the loop at the end.
That is exactly what it does. Dicer1 is another RNase enzyme. It grabs the base of the hairpin, measures up the stem a very specific distance, usually about 21 to 25 nucleotides, and slices off the loop.
So it's flat on both ends now.
Yes. What you are left with is no longer a hairpin. It is a perfectly sized, double-stranded RNA molecule called an shRNA duplex.
So the hairpin has been chopped into 2 separate parallel strands. And this is where the hitman finally gets his weapon loaded. This duplex gets handed over to a massive multi-protein machine called the RISC, the RNA-induced silencing complex. But the research says RISC can't use both strands of the duplex. It has to throw one away.
That is correct. RISC needs a single solitary guide strand to go out and find the target. The main engine of the RISC complex is an incredibly complex protein called Argonaute-2, or AGO2.
AGO2 is the hitman.
AGO2 is the actual assassin. But when it receives that double-stranded duplex, it has to decide which strand to keep as the guide strand and which strand to discard as the passenger strand.
Wait, how does a blind protein know which strand is which? I mean, it's just molecular machinery floating in fluid.
It makes a thermodynamic choice. AGO2 physically tests the stability of both ends of the duplex. Remember, the 2 strands are held together by hydrogen bonds between the base pairs.
Right.
Certain base pairs, like guanine and cytosine, have 3 hydrogen bonds and are very tight. Adenine and uracil only have 2, making them looser. AGO2 tests the ends and preferentially picks the strand whose 5' end is less thermodynamically stable.
A looser end.
Exactly, the looser end. It locks onto that loose end, pulls the strands apart, keeps the guide strand, and literally shreds the passenger strand, degrading it completely.
It's ruthless. It just tests them, keeps the one that's slightly looser, shreds the other one, and now it has its guide. It has the photograph of the target.
Armed with that single guide strand, AGO2 scans the cytoplasm. It bounces around, testing every messenger RNA it bumps into against the sequence of its guide strand. When it finds an mRNA that perfectly 100% matches the sequence of the guide strand, AGO2 changes its physical shape.
Wow.
It acts like a molecular scalpel. It slices the target mRNA exactly between nucleotides 10 and 11 relative to the guide strand.
Wait, exactly between nucleotides 10 and 11. That level of physical precision is mind-blowing. Once it's sliced, what happens to the target?
Once the mRNA is cut in half, it loses its protective caps. The cell's natural cleanup enzymes immediately recognize it as garbage and rapidly degrade the rest of it into single nucleotides.
So it's just recycled.
Exactly. The blueprint for the disease-causing protein is completely destroyed before the cell's ribosomes ever have a chance to build the protein.
Okay, I have to stop and ask a big-picture question here. Why on earth does a human cell have this incredibly complex, multi-step machinery, Grosha, exportin 5, Dicer 1, IRTC, Argonaut 2, just sitting around waiting to be used? We didn't evolve these machines specifically to process pharmaceutical drugs, right?
No, we didn't. That is a fundamental question. The cell didn't evolve this machinery for our benefit. We are essentially hijacking an ancient natural cellular defense system.
Hijacking it.
Yes. This pathway evolved millions of years ago in our evolutionary ancestors. Originally, it was an immune response to protect organisms from double-stranded RNA viruses.
Like a viral defense system.
Precisely. Many ancient viruses injected double-stranded RNA into cells. The cell evolved Dicer to chop up that viral RNA and RISC to use those pieces to hunt down the rest of the virus.
That makes so much sense.
Over millions of years, the cell also adapted this machinery to manage its own internal gene regulation through naturally occurring microRNAs. So when we use shRNA as a drug, we are just slipping our own synthetic, highly targeted instruction manual into an ancient defense system that is already humming along inside every single one of your cells.
That makes a lot of sense. But going back to our factory analogy for a second, what happens if the instructions are slightly off? What if Dicer 1, the quality control inspector, cuts the loop a little too high or a little too low? If the cut is messy, doesn't that change the sequence of the guide strand?
It does, and that is one of the most significant challenges in RNA interference therapy. If Dicer 1's cut is imprecise, it alters the specific sequence of nucleotides that ends up loaded into AGO2. In the scientific literature, this is called a seed shift.
A seed shift. Returning to the hitman analogy, does that mean the hitman is suddenly handed the wrong photo?
Precisely. The seed region of the guide strand, which is nucleotides 2 through 8, is the most critical part for recognizing the target. If Dicer 1 cuts the hairpin poorly, that seed sequence shifts.
Oh.
Instead of taking out the disease-causing gene, the RISC complex might now perfectly match a vital innocent bystander gene. Your own internal machinery starts slicing up the mRNAs that keep your cell alive.
Extremely fire.
Exactly. That off-target cleavage can lead to massive cellular toxicity and cell death.
Okay, so we've mapped out the factory. We know how the hitman gets the photo, how the thermodynamic choices are made, and how the cut happens at nucleotide 10 and 11. But there's a glaring issue here. All of this relies on getting the blueprint, that DNA vector you mentioned at the very beginning, inside the cell's tightly guarded nucleus in the first place.
The delivery problem.
Yes. For a living, breathing patient, how do we smuggle the shRNA blueprints past the cell membrane, through the cytoplasm, and into the nucleus?
That is known broadly as the delivery problem, and it is arguably the single greatest hurdle in all of genetic medicine. You can have the most beautifully designed shRNA in the world, but if you can't get it into the target tissue, it's useless. There are two primary battlegrounds here. Systemic delivery, where you infuse the therapy into the patient's bloodstream and try to target a specific internal organ, and ex vivo delivery, where you physically extract cells from the patient's body, modify them in a lab where you control the environment, and then infuse them back into the patient.
Let's start with systemic delivery, because looking at the research, it mentions using viruses as delivery trucks. I'm going to push back on this. Injecting a stripped-down virus into a patient's bloodstream to cure disease sounds terrifying. If I'm a patient and you tell me you're going to give me a virus to cure me, my immediate thought is that my immune system is going to go crazy.
Your reaction is completely natural, and it's the exact same reaction the immune system has. But viruses are the ultimate delivery vehicles. They have spent billions of years perfecting the art of breaking into cells, evading initial defenses, and delivering genetic payloads into the nucleus.
So we just steal their technology.
Exactly. Bioengineers take these viruses, completely hollow them out by removing all of their native disease-causing replication genes, and replace that internal machinery with our therapeutic shRNA blueprints. The gold standard for this right now are recombinant adeno-associated viruses, or AAVs.
Why AAV specifically? I mean, what makes them better than, say, a common cold virus?
AAVs are incredibly safe. They don't typically cause disease in humans, and importantly, they don't integrate their payload directly into the host's chromosomes, which reduces the risk of accidental mutations. Furthermore, they have a property called tropism.
Tropism.
Tropism means that the physical proteins on the outside of the virus naturally bind to specific receptors on specific types of human cells. Different AAV serotypes naturally gravitate toward entirely different tissues.
Oh, so it's like having a delivery truck that is mechanically programmed to only drive to one specific neighborhood, ignoring all the other houses on the way.
That's a perfect way to look at it. Let's look at the data. AAV8 has a very high natural tropism for the liver. It binds beautifully to hepatocytes. The research highlights clinical trials using AAV8 to deliver shRNA for hepatitis C.
So it just goes straight to the liver.
Right. The goal is to infuse the AAV8 into the bloodstream, let it naturally accumulate in the liver, and turn those liver cells into localized shRNA factories that continuously produce assassins to hunt down the hepatitis C viral RNA.
Wow.
On the other hand, AAV9 has a strong natural affinity for muscle cells and neural tissue. It can even cross the blood-brain barrier to some extent, which makes AAV9 invaluable for treating muscular dystrophies or neurodegenerative diseases.
Okay, but I still have to wonder about the immune response.
Yeah.
Even if it's hollowed out, the outside of the virus, the shell, is still made of viral proteins, right? Isn't the patient's body going to recognize those proteins, create neutralizing antibodies, and destroy the delivery trucks before they can drop off the blueprints?
You've hit on the critical limitation of viral vectors. Yes, the immune system will eventually recognize the viral capsid. Once a patient is dosed with an AAV, their immune system generates massive amounts of neutralizing antibodies against that specific virus.
So that's a one-time thing.
Usually, yes. This means you generally can only dose a patient with the specific AAV serotype once in their entire life. If you try to give them a second dose a year later, the immune system will instantly destroy the virus before it ever reaches the target organ.
Wow. So if the disease requires redosing, you're just out of luck with AAVs.
Yeah.
I assume that's why I'm seeing an entirely separate branch of research in our stack focused on non-viral vectors, synthetic carriers.
Exactly.
There's a trial here that really caught my eye, the STMN1 BIV trial. It says they were injecting advanced refractory solid tumors with something called bilamellar invaginated vesicles. That is a massive mouthful. What is a bilamellar invaginated vesicle?
Let's break that down. A vesicle is essentially a tiny microscopic bubble. Bilamellar means it has a double layer of lipids, or fats. And invaginated means those layers are folded inward upon themselves, creating complex internal compartments that protect the fragile DNA plasmid inside.
Okay, a folded fat bubble.
Right. In the STMN1 trial, they built this synthetic lipoplex out of two main ingredients: DOTAP, which is a positively charged lipid, and cholesterol.
Wait, why cholesterol? We usually think of cholesterol as a bad thing that clogs arteries.
In biochemistry, cholesterol is a structural component. It adds rigidity and stability to lipid membranes. When you inject these synthetic fat bubbles into the chaotic, high-shear environment of the human bloodstream, they need to survive forces that would otherwise rip them apart.
Ah, so it's armor.
The cholesterol acts like scaffolding. Now, because this bubble is entirely synthetic and not a virus, the immune system's radar doesn't immediately ping it as a biological pathogen.
But the body still clears it out eventually, right? The notes on the pharmacokinetics for the STMN1 trial are wild. It says that when they injected these synthetic lipoplexes, the amount of circulating DNA plasmid in the patient's blood peaked at just 30 seconds. And within 5 minutes, it plummeted to less than 10%.
Where did it go? Did it just fall apart?
No, it didn't fall apart. It was rapidly internalized. A large portion was quickly taken up by the cells in the target area, while the rest was filtered out by the reticuloendothelial system.
Which is what?
The body's natural clearance organs, like the liver and spleen. But this rapid clearance is actually a massive advantage.
Really? Why is disappearing quickly a good thing? I would think you'd want the drug to stick around.
Because it minimizes systemic immune flare-ups. The longer a foreign substance, even a synthetic lipid, circulates freely in the bloodstream, the higher the chance it triggers a massive systemic inflammatory response.
Oh, I see. By designing the lipoplex to be taken up almost immediately, they drastically narrow the window for the immune system to react. In the SDMN1 trial, this rapid pharmacokinetic profile allowed them to successfully deliver the blueprints into the tumor cells, achieving cleavage of the target mRNA without triggering the severe, life-threatening immune reactions that often plague gene therapies.
That is brilliant engineering. Use the body's rapid clearance against it to sneak the payload in quickly. But the source has also detailed a delivery method that sounds even stranger than synthetic fat bubbles. Reconstituted biological carriers. It says they're using influenza virus envelopes.
Yes, this represents a fascinating middle ground. It's a hybrid between viral and non-viral delivery. Researchers asked, what is the single hardest part of getting into a cell? It's not getting swallowed by the cell membrane, it's escaping the endosome.
The endosome. Let's define that for anyone listening who might not be familiar. When a cell swallows something from the outside, it doesn't just dump it directly into the cytoplasm, right? It traps it in a little internal bubble called an endosome.
Exactly. The endosome is like a sorting room, but it's also a digestion chamber. As soon as the endosome forms, the cell starts pumping protons into it, rapidly dropping the pH and making the environment highly acidic.
Like a stomach.
Exactly like a stomach. This acid activates digestive enzymes that shred whatever the cell just swallowed. If your therapeutic lipoplex is trapped in the endosome, your drug is destroyed.
So how does the influenza virus envelope help with that?
Researchers figured out how to extract a very specific protein from the surface of the influenza virus, the hemagglutinin protein. They strip away everything else about the flu virus, taking only this one protein, and embed it into their synthetic lipid carrier.
Okay.
Hemagglutinin is nature's ultimate lockpick. It is a highly specialized, pH-sensitive protein. When the cell swallows the carrier and the endosome becomes acidic, the drop in pH causes the hemagglutinin protein to undergo a massive physical shape change.
It reacts to the acid.
Yes. It physically snaps open like a spring, exposing a highly hydrophobic fusion peptide that violently stabs into the membrane of the endosome. It forces the endosomal membrane to fuse with the lipid carrier, literally ripping a hole in the endosome. This forces the endosome to burst open, dumping the fragile shRNA cargo safely into the cytoplasm before the acid and enzymes can digest it. And it does all of this purely through biomechanical reactions to pH without using a live replicating virus.
I love that. It's like dressing your synthetic fat bubble in a highly specific disguise just long enough to get past the internal security guards. And the moment the guards try to throw it in the incinerator, the disguise violently explodes and the payload drops through a trapdoor. Amazing.
It's incredible biological machinery.
Okay, so we've established the factory inside the cell, and we figured out how to smuggle the blueprints in through AAVs or lipid carriers. Now let's look at the absolute peak of this technology's precision. What happens when the target isn't a foreign invader like hepatitis C or a massive tumor, but a single microscopic spelling error in the patient's own DNA? Let's talk about allele-specific silencing.
This specific application is where shRNA truly shines and demonstrates capabilities that almost no other therapeutic modality can match. Let's ground this in a real-world example. Autosomal dominant genetic diseases, like Huntington's disease.
I want to spend some time on this because Huntington's is a horrific disease. It's a progressive, fatal neurodegenerative disorder. It robs people of their motor control, their cognition, their personality. And the genetics of it are particularly cruel.
They are very cruel. In Huntington's disease, a patient inherits 2 copies of the HTT gene 1 from each parent. Typically, they inherit one perfectly healthy wild-type copy from one parent and one mutated copy from the other parent.
Okay.
Because it is a dominant disease, that single mutated copy is enough to cause the entire illness.
And looking at the research, the mutated copy has something called a polyglutamine expansion. It's basically a stutter in the genetic code, right?
Yeah.
A repeating KEGG sequence that just goes on for too long.
That's a great way to visualize it. The DNA stutters. When the cell transcribes that stuttering DNA into mRNA, and the ribosome translates that mRNA into a protein, it produces a toxic, misfolded HTT protein.
And that builds up.
Yes, this toxic protein clumps together, aggregates, and physically destroys the striatal neurons in the brain over time.
Okay, I'm playing devil's advocate here. If we have an shRNA hitman, and we know the target is the HTT messenger RNA, why don't we just design a guide strand that binds to the KEGG stutter and destroys it? Or better yet, just shut down the entire factory and destroy all the HTT mRNA.
That is the obvious first thought, but it would be catastrophic for the patient. Let's start with the first idea. Targeting the CAG stutter. You cannot target the CAG repeat because that specific sequence exists in dozens of other vital, healthy genes throughout the genome.
Oh, I see.
If you send an assassin looking for CAG, it will destroy the Huntington's mutation, but it will also destroy numerous healthy proteins your brain needs to survive. It's a carpet bomb.
Right, the off-target toxicity we discussed earlier. So what about the second idea? Just shutting down the whole HTT gene entirely, both copies.
You can't do that either. The wild-type, healthy HTT protein produced by the normal gene is absolutely vital for normal brain function and neuronal transport. If you blindly destroy both the healthy and the mutated versions of the mRNA, you induce massive transcriptomic changes.
So you cure the Huntington's but kill the neuron anyway.
Exactly. The neurons might survive the lack of the toxic protein, but they will likely die from the lack of the healthy protein. You have to thread an incredibly fine biological needle. You need a mechanism to destroy the mutated copy while leaving the healthy copy completely 100% untouched.
And how on earth do you do that when the two messenger RNAs are essentially 99.9% identical? I mean, they're produced by the exact same gene, just from different alleles.
You stop looking at the stutter, and you look for the 0.1% difference elsewhere on the mRNA strand. Researchers painstakingly analyzed the entire genetic sequence of both alleles, looking for SNPs single nucleotide polymorphisms.
I've heard of SNPs in the context of, like, ancestry DNA tests.
Yeah.
But what are they doing here?
A SNP is simply a tiny, naturally occurring single-letter variation in the genetic code. It's a minor spelling difference that doesn't usually affect the function of the protein. By analyzing patients, researchers found specific SNPs that happen to physically sit on the mutated, disease-causing allele, but do not exist on the healthy allele.
Okay.
The SNP is physically linked to the mutation.
Okay, this is incredible. So it's not a carpet bomb. It really is a molecular sniper. It's like looking at a lineup of identical twins. They look exactly the same. But you know, the evil twin has a single tiny freckle on their left cheek. The SNP is the freckle. You train the hitman to only shoot if he sees the freckle.
That is an exceptionally accurate analogy. The research focused on targeting SNPs located in specific sections, or exons, of the HTT genes, specifically exons 39, 50, 60, and 67. The bioengineers built chimeric shRNAs where the sequence of the guide strand perfectly 100% matched the SNP of the mutated gene. But critically, that meant the guide strand had a one-letter mismatch with the healthy gene.
Because remember, earlier we talked about how the hitman needs a perfect photo. Argonaut 2 slicing is dependent on thermodynamic stability. If there is a single-letter mismatch, the geometry of the RNA helix bulges and the stability is thrown off.
Precisely. If there is a bulge caused by a mismatched base pair, AGO2 cannot achieve the correct physical conformation to make the cut at nucleotides 10 and 11. It's physically impossible.
It just lets go.
It does. They tested this elegant concept in human embryonic stem cells, in rat models, and in highly advanced transgenic mice known as BACHD mice, which are engineered to express the full-length human mutant Huntington's gene. The empirical results were staggering.
What happened when they ran the tests?
The mismatched shRNAs, the ones encountering the healthy gene without the SNP, caused almost zero degradation. The healthy HTT mRNA levels remained completely stable, but the matched shRNAs, the ones that found the SNP freckle locked onto the toxic mutant mRNA, achieved the perfect thermodynamic fit and allowed AGO2 to rapidly clear it out. They isolated the evil twin and eliminated it.
And I want to emphasize, reading these notes, it wasn't just a chemical victory in a petri dish. There was actual measurable functional recovery in the neurons, right?
Yes. In the neurons derived from human stem cells, researchers tracked the movement of BDNF brain-derived neurotrophic factor. BDNF is a crucial protein that helps neurons survive and grow. It moves up and down the long arms of the neuron along microtubule tracks.
Like a train on a track.
Exactly. In Huntington's, the toxic mutant protein physically clogs up these tracks, blocking BDNF transport. By using allele-specific shRNA to clear out the toxic mutant HTT protein, they successfully restored the vesicular transport of BDNF along the microtubules. The neurons literally began to heal and function normally again because the physical blockade was removed, while the healthy HTT protein continued its vital daily work.
That is just brilliant. You restore the physical highway of the cell, And Huntington's isn't the only example of this sniper approach in the sources. They also talk about a condition called long QT1 syndrome.
Long QT1 is a severe cardiac condition that causes very dangerous, sometimes fatal arrhythmias. It is driven by a dominant mutation in a gene called KCNQ1, which controls the potassium channels in the heart. Just like in Huntington's, patients have a mix of healthy and mutant proteins actively working in their heart cells, competing with each other and causing chaos in the electrical signaling.
And the researchers use the same freckle hunting technique.
Exactly. In the trial, they utilized an allele-specific shRNA called HA18. This shRNA was perfectly matched to a SNP on the mutant allele. They targeted the heart cells and were able to selectively knock down the production of the mutant potassium channel. What's fascinating here is the shift in the cellular balance.
What was the balance?
Before the treatment, the ratio of healthy protein to mutant protein in the cells was a dangerous 43 to 57. The mutant was winning. After treating the cells with the shRNA18 shRNA, that ratio dramatically flipped to a healthy 63 to 37.
You don't even have to eliminate it completely. Just by tilting the scale back in favor of the healthy protein, you stabilize the electrical signaling and stop the arrhythmias.
Okay.
But I have to be realistic here. This sniper approach sounds perfect, almost too perfect. What is the catch with allele-specific silencing?
The primary catch is scalability and patient coverage. Because every single patient's genetics are slightly different, the specific SNP freckle that accompanies one patient's Huntington's mutation might be completely different from the SNP found in the next patient's DNA.
Right.
One drug will not cure everyone. To theoretically treat the majority of the Huntington's population, epidemiological researchers estimate you might need to develop, extensively test, and individually approve a library of 8 or more entirely different shRNA drugs.
So you have to genetically sequence the patient, find their specific SNP, go to the library, and pull the exact shRNA that matches their unique genetic fingerprint. It is personalized medicine taken to the absolute extreme, which I imagine presents massive regulatory and financial hurdles.
Enormous hurdles. Getting one gene therapy approved costs hundreds of millions of dollars. Getting 8 slightly different versions approved for the exact same disease is an economic nightmare under current regulatory frameworks.
Okay, so that perfectly sets up my next major question. If allele-specific silencing is so incredibly perfect sniper-like precision, saving the healthy gene, but it relies on finding these unique patient-specific SNPs, what happens if the genetic mutation is just too tricky? What if the mutation is too small or it's standard across all patients without a unique distinguishing SNP nearby? Are those patients just out of luck? I mean, why aren't we using the sniper approach for everything?
When you can't use a sniper, you have to bring in a demolition crew and an architect simultaneously. This leads us to the silence and replace strategy.
Silence and replace. It sounds like a home renovation show, but for DNA.
The home renovation analogy is actually very fitting. The quintessential example detailed in our sources is the BB301 clinical trial for a devastating disease called oculopharyngeal muscular dystrophy, or OPMD. OPMD causes severe progressive dysphagia, meaning the muscles in the throat weaken over time, and the patients slowly lose the physical ability to swallow food or liquid.
That sounds awful.
It is a terrible way to suffer. It's driven by a mutation in a gene called PBPN1.
And I'm guessing they couldn't find a convenient SNP freckle for PBPN1.
Precisely. Finding a reliable, widely applicable allele-specific SNP for OPMD proved incredibly difficult due to the specific nature and location of the mutation. The sniper approach wasn't viable, so the researchers behind BB301 took a completely different, radical approach.
How does the BB301 drug actually work then, if it can't distinguish between the healthy and mutant alleles?
They deliver a single AAV9 viral vector directly into the pharyngeal muscles of the patient's throat. But this specific viral vector is engineered to carry 2 distinct genetic payloads. The first payload is a highly potent, completely non-selective shRNA.
Non-selective.
Yes, it doesn't look for a freckle. It doesn't care about SNPs. It is designed to aggressively target and destroy every single copy of the PABPN1 messenger RNA it finds, both the mutant, disease-causing ones and the healthy, wild-type ones. It completely levels the entire house to the foundation, wipes the slate clean.
Hang on, I'm losing you. If you completely wipe out all of the PADPN1 protein, the muscle cell is going to die. You just told me 5 minutes ago with Huntington's that destroying the healthy protein is catastrophic.
It would be catastrophic, which is why that exact same AAV9 vector carries a second payload. The second payload is a brand new, pristine, healthy replacement gene. The architect arrives exactly as the demolition crew finishes, but this replacement gene is a codon-optimized version referred to in the trial as CoPAB-PN1.
I feel like I'm missing something fundamental here. If the cell is pumping out this highly aggressive, non-selective shRNA hitman that is actively destroying all PAPN1 messenger RNA in the cytoplasm, how does the replacement gene's mRNA survive? Why doesn't the hitman just turn around and shoot the new architect? Is it physically shielded somehow?
There is no physical shield. It's a brilliant linguistic trick. It relies on the inherent redundancy built into the human genetic code. Let's look at the biophysics of translation. Multiple different three-letter DNA codes, which are called codons, can produce the exact same amino acid.
Okay, I follow that.
For example, the amino acid leucine can be coded by 6 completely different three-letter DNA sequences.
So there are multiple ways to spell the same word.
Exactly. Scientists computationally altered the spelling of the replacement gene. They swapped out the native codons for alternative codons that produce the exact same amino acids. This codon-optimized gene produces a healthy 100% functional PAPN1 protein that the muscle cell can use to survive and rebuild. But the underlying messenger RNA sequence is spelled so drastically differently that the shRNA hitman floating in the cytoplasm doesn't recognize it. The guide strand cannot achieve a thermodynamic match with the new spelling. The shRNA completely ignores the replacement mRNA, allowing it to translate into the cure.
That is just, it's a biochemical magic trick. You tear down the diseased house, keep the family alive, and simultaneously build an identical pre-built house on the exact same lot, but you paint it a completely different color so the demolition crew ignores it.
Yeah.
And the clinical results for BB301 in human patients were stunning, right?
Unprecedented. In cohort 1 of the clinical trials, observing patients with this severe muscular dystrophy, they saw a 100% responder rate. Patients who had been steadily losing the ability to swallow experienced durable, stable improvements in their swallowing function at both 12 and 24 months post-treatment.
That is incredible.
And crucially, there were no severe treatment-related adverse events. They halted the progression of a previously untreatable disease.
Okay, I have to ask, if we can just wipe the slate clean and install a completely new stealth operating system like this, why do we even bother with the sniper approach at all? If silence and replace works so perfectly for OPMD, why not just silence and replace everything? Why not do it for Huntington's?
Because you have to consider the metabolic burden on the patient's cells. Silence and replace is highly effective, but the biological toll on the cell is immense. The cell is being forced to constantly, 24/7, manufacture the destructive shRNA, process it through exportin 5 and dicer1, load it into RISC, and simultaneously use its transcription and translation machinery to constantly pump out the synthetic replacement protein, all driven from a single artificially introduced vector.
It's forcing the cell to work massive amounts of overtime.
Exactly. For a relatively small, localized tissue mass, like the pharyngeal muscles in the throat for OPMD, the cells can handle that workload. It works beautifully. But imagine trying to sustain that level of synthetic overproduction across the entire mass of the human brain for Huntington's disease, or across a massive, highly active organ like the liver.
It would just burn out.
Yes. You risk pushing the cells into profound metabolic exhaustion. The elegant precision of the allele-specific sniper approach, when it is possible, is vastly gentler on the host organism because it relies on the cell's natural expression of the healthy gene.
That makes perfect sense. It's a sledgehammer versus a scalpel. Okay, we've solved single gene mutations. Huntington's, long QT1, OPMD, those are relatively straightforward in concept. Let's escalate. Let's look at complex systemic diseases like cancer, where it's never just one simple spelling error. Cancer is a chaotic cascade of multiple things going wrong simultaneously. Can shRNA handle multiple targets at the exact same time?
Yes, it can, through an incredibly powerful structural concept called multiplexing. And the prime example in the scientific literature provided is the CYA2111 clinical trial for relapsed and refractory multiple myeloma.
Multiple myeloma is a brutal, relentless blood cancer. It attacks the plasma cells in the bone marrow, often causing the bones themselves to literally dissolve and break. But reading these notes, CYAD211 isn't just an shRNA drug infused in a lipid bubble. It's an entirely different class of medicine. It's a CAR T-cell therapy.
That's right. CAR T-cell therapy is one of the greatest breakthroughs in oncology. Normally, it involves extracting a patient's own immune T-cells engineering them in a lab with a chimeric antigen receptor, a CAR, to recognize and kill the cancer, and infusing them back into the patient. But that autologous process is agonizingly slow, incredibly expensive, and sometimes the patient's cells are already too damaged by chemotherapy to be useful.
So what makes CYE211 different?
CY8211 aims for the holy grail of cell therapy, allogeneic CAR-R T cells. This means creating an off-the-shelf therapy using healthy T cells from a completely different healthy donor. You engineer them in massive batches and freeze them, ready for any patient who walks in the door.
But if you put someone else's donor T cells into my body, won't those donor cells immediately recognize my body as a foreign invader and start attacking my organs?
Yes, that is a fatal condition known as graft versus host disease, or GVHD. The donor T cells look at the patient's tissue and attack. So how does shRNA fix that? By multiplexing. In CY8211, bioengineers use a single shRNA genetic platform designed within a very specific microRNA scaffold to knock down up to 4 entirely different target genes in the donor T cells simultaneously, all from one transcript.
Wait, 4 different targets from one blueprint. What are they knocking down?
First, they knock down the CD3 zeta subunit. This is the receptor that allows the T cell to recognize foreign tissue. By silencing it, they effectively blind the T cell to the patient's healthy tissue, completely preventing GVHD.
Okay, that solves the friendly fire issue.
But they don't stop there. They also knock down genes like B2M and c-IDA. Silencing these helps the donor cells evade the patient's own immune system, allowing them to persist in the body longer to fight the cancer. And fourth, they knock down a gene called PD-1.
I recognize PD-1. That's an exhaustion marker, right?
Yeah.
Cancer cells often exploit PD-1 to put T cells to sleep.
Exactly. Tumors express a protein that binds to PD-1, essentially hitting the off switch on the T cell. By multiplexing an shRNA against PD-1, the researchers removed the off switch. They are custom engineering an unstoppable supersoldier cell by suppressing 4 different cellular vulnerabilities at once, driven entirely from a single genetic construct.
But wait, why use hRNA for this? If we're engineering these cells in a lab anyway, everyone talks about CRISPR for gene editing. Why not just use CRISPR to permanently snip out those 4 genes from the DNA?
That is a critical distinction in the field of gene editing. If you use a CRISPR-like tool to physically cut the DNA genome in 4 entirely different places at the exact same time, you drastically increase the risk of a catastrophic event called chromosomal translocation.
What does a chromosomal translocation mean physically for the DNA inside the nucleus?
Think about the DNA as a library of encyclopedias. CRISPR works by making double-strand breaks. It literally chops the books in half. If you make 4 breaks at once, you have loose ends of chromosomes floating around in the nucleus.
That sounds dangerous.
It is. When the cell's emergency repair machinery frantically tries to stitch the double-strand breaks back together, it can get confused. It might accidentally stitch a piece of chromosome 1 onto a piece of chromosome 4.
That sounds like a recipe for a massive oncogenic mutation.
It is. You can inadvertently create new cancers in the very cells you are engineering to fight cancer. shRNA avoids this entirely. shRNA only targets the transient messenger RNA floating out in the cytoplasm. The master DNA genome inside the nucleus remains perfectly intact, completely uncut, and entirely safe from translocations.
That is a massive safety advantage for hRNA. But cancer multiplexing gets even wilder. Let's look at the VIGIL trial, also known as Gemo-Genevetocel-T. This was for advanced ovarian cancer. The notes describe this as using a bifunctional hRNA. What does bifunctional mean in this context? Because so far we've just talked about the hitman slicing the mRNA.
This represents one of the most structurally innovative biochemical approaches in the entire field. A bifunctional hRNA doesn't just rely on the standard AGO2 slicing we've been discussing. In the VIGIL trial, the plasmid delivered to the tumor cells encodes an RNA molecule that forms 2 distinct stem loop structures simultaneously.
Okay, 2 loops. What do they do?
The first loop is designed as a perfect 100% thermodynamic match to the target mRNA. This drives the classic cleavage-dependent destruction by AGO2, slicing it in half. But the second loop on that exact same molecule is intentionally designed by the engineers with a specific mismatch in its sequence.
A mismatch. But we established earlier that a mismatch prevents AGO2 from making the cut. Why would you intentionally design a broken hitman?
Because of how the RISC complex handles a mismatched guide strand, if the guide strand isn't a perfect match, AGO2 cannot physically slice the mRNA. But it doesn't just let go. Instead, it triggers a completely different mechanism called cleavage-independent loading or translational repression. The RISC complex physically sits down on top of the target messenger RNA and parks there.
Like a roadblock.
exactly like a physical roadblock. When the cell's massive ribosome machinery comes along the mRNA track trying to read it and build a protein, it slams into the RASC complex and gets stuck. It jams the translation machinery entirely.
Wow.
By utilizing a bifunctional vector that triggers both rapid cleavage and relentless translational repression simultaneously, researchers achieve a highly robust 90 to 95% knockdown of the target protein. It is a dual-pronged attack that almost entirely eliminates the chance for the cancer cell to mutate its RNA sequence and escape the therapy.
That is brilliant. You cut the supply lines, and whatever supplies remain, you set up a barricade so they can't be used.
That it.
And in the Vigil trial, what exact protein are they hitting with this bifunctional attack?
They are targeting a gene that produces an enzyme called furin. Furin is a protease. Its job is to process another molecule called TGF-beta into its highly active immunosuppressive form. Ovarian cancer tumors are notoriously difficult to treat because they aggressively pump out active TGF-beta into their microenvironment.
Like a force field.
Exactly. It
Yeah.
creates a localized force field. It physically blinds the patient's natural immune system to the presence of the tumor.
So by knocking down furin with this bifunctional shRNA, you prevent the creation of TGF-beta, and the force field drops. The invisibility cloak is removed.
Correct. But Vigil goes one step further. While the hRNA knocks down furin, the exact same plasmid also forces the tumor cell to express and pump out a highly visible immune-stimulating protein called GM-CSF.
So let me get this straight. The drug strips away the tumor's invisibility cloak and simultaneously sets off a massive alarm siren inside the tumor, screaming for the immune system to come look.
Precisely. GM-CSF actively recruits specialized immune troops dendritic cells and cytotoxic T lymphocytes directly to the physical site of the tumor. You drop the defenses and invite the attack. The dendritic cells scoop up the newly exposed cancer neoantigens, process them, and train the rest of the immune system to hunt the cancer systemically.
That is incredibly clever.
And the clinical data reflects the brilliance of this mechanism. In patients with wild-type BRCA advanced ovarian cancer, the relapse-free survival improved from 14.8 months in the control group to an astounding 19.4 months in the Vigil group.
An extra 4.5 months of relapse-free survival in advanced ovarian cancer. That is massive for those patients. But okay, I have to pause. I'm listening to all this and it sounds like pure magic. We are building microscopic thermodynamic snipers. We are engineering super soldier T cells that don't get exhausted. We are dropping stealth tumor alarms. It's perfect.
So what is the catch? I mean, why isn't every single genetic disease on Earth being cured by shRNA right now? We have to look at the dark side of this technology. The dark side is very real, and it has humbled the field of gene therapy more than once. To understand why, we have to go all the way back to the factory analogy we started with. The cellular machinery exportin 5, dicer 1, the ISC complex, these are not infinite resources.
Right. There's only so many to go around.
A cell only has a certain number of these machines available at any given time. Early researchers, filled with optimism, assumed a linear relationship. More shRNA drug meant a stronger knockdown and a better cure. So they pumped massive amounts of highly active shRNA viral vectors into mice.
I have the paper right here, the Grimm et al. Study from 2006. This was a massive wake-up call for the entire industry. What exactly did they do to these mice?
Grimm and his colleagues delivered AAV vectors expressing various shRNAs into the livers of mice. But they used incredibly strong promoters flooding the cells with synthetic hairpin RNA. What they discovered was fatal oversaturation.
Too much of a good thing.
Far too much. If you flood the cell with millions of synthetic shRNA hairpins, they hog all of the exportin 5 transport bouncers at the nuclear pore. They saturate all of the dicer 1 enzymes and RASC complexes. The synthetic drug completely takes over the factory floor.
And why is that fatal?
Right.
If the factory is just making the cure faster, why does the cell die?
Because the cell relies on that exact same factory machinery to process its own naturally occurring microRNAs. Natural microRNAs are absolutely essential for survival. They regulate the cell cycle, they manage stress responses, they control apoptosis or program cell death. When the synthetic drug hogs machinery, the natural microRNAs can't get processed.
They back up.
Right. Without those natural regulators managing the day-to-day operations, the entire cellular ecosystem collapses into chaos. In Grimm's 2006 study, this competitive oversaturation led to fatal hepatotoxicity, massive, acute liver failure that killed the mice.
It's literally putting so many priority rush orders into the factory that the workers can't do basic maintenance, and the entire factory just burns to the ground. And reading further, it's not just the liver that's sensitive. The sources detail neurotoxicity in the brain too. And this experiment is honestly terrifying. It's the Schlufik experiments. It says they ran an experiment in rats where they used a completely harmless control shRNA.
Ah, yes, the Schlufik experiments. Researchers designed a specific shRNA intended to target the luciferase gene, the gene that makes fireflies glow. Now, obviously, rats do not have firefly DNA in their brains.
Of course not.
So introducing this Schloop shRNA should have done absolutely nothing. It was intended to be a blank, negative control to prove the delivery system was safe.
But the notes say it wasn't a blank, it caused severe damage.
Far from blank. When researchers injected this supposedly harmless, non-targeting shRNA into the hippocampus of the rat brains, they observed severe neurotoxicity. Under the microscope, they literally watched the synapses and dendritic spines, the vital communication branches that allow neurons to talk to each other, physically retract and wither away.
Why? I don't understand. If there is no firefly gene for the RISC complex to cut, what was AGO2 actually doing in those rat neurons?
It comes back to the seed region of the shRNA guide strand. Even though the full 21 nucleotide sequence of the Schluka drug didn't match anything in the rat genome, a tiny, short stretch of that sequence, just 6 or 7 nucleotides in the seed region, accidentally, purely by chance, formed a partial match with several native messenger RNAs that were critical for neuronal maintenance and synaptic growth.
Just a partial match of 6 letters was enough.
It was enough of a thermodynamic match for the RISC complex to start binding to those vital neural genes and initiating translational repression, exactly like the visual trial did on purpose, but here it was an accident. The drug inadvertently starved the neurons of the proteins they needed to maintain their connections. It highlights how exquisitely sensitive the mammalian brain is to even the slightest off-target RNA interference.
Okay, so what does this all mean for the field? If the drug is too efficient, it hogs the factory and kills the liver. If it has a tiny 6-letter random match, it withers the brain. How do bioengineers fix a drug that is essentially too strong and too aggressive for its own good?
Fixing it requires a profound, counterintuitive shift in how we approach pharmacology. The TT-034 clinical trial for hepatitis C perfectly illustrates this modern approach. To prevent the fatal liver toxicities seen in the 2006 Grimm models, the scientists at Tessir Therapeutics realized they couldn't just pump in maximum doses. They did something highly unorthodox. They actively, intentionally weakened the genetic machinery of the drug.
They weakened it. How do you physically weaken a DNA blueprint?
Think back to the very first step we discussed, the Pol3U6 promoters, the molecular engines that drive the transcription of the blueprint. Tessir bioengineers went into the DNA code of the U6 promoter and intentionally mutated its proximal sequence elements. They swapped out the optimal, high-efficiency binding sites for weaker, suboptimal versions. This created hybrid, deliberately crippled promoters, which they named U67 and U68.
Why cripple the engine?
By crippling the engine, the RNA polymerase machinery couldn't bind as tightly or transcribe as quickly. They drastically slowed down the rate at which the shRNA rolled off the assembly line. Instead of flooding the factory with millions of hairpins and overwhelming export in 5, the weakened promoters just sent a steady, low-level, manageable trickle of drug into the cytoplasm.
And a trickle is enough to kill the virus.
It was. The steady trickle produced just enough shRNA to successfully target and cleave the hepatitis C viral RNA. But crucially, it kept the overall synthetic RNA levels low enough that the liver cell's exportin 5 and RISC machinery could comfortably process both the drug and the cell's own vital natural microRNAs. Biology is ultimately about homeostasis, about maintaining balance. TT-034 respected that cellular balance instead of trying to overwhelm it.
That is an incredible pivot, but looking at the notes for the TT-034 trial, story gets even weirder. There is a section here labeled the deep sequencing anomaly. What happened there?
When TT-034 went into trials, researchers used next-generation deep sequencing to analyze the actual RNA strands present inside the treated liver cells. Now, we've discussed Dicer1 as a precision quality control inspector, always cutting exactly at the same base pair to produce one specific, predictable guide strand.
Right.
But when Tacer analyzed the cells, they found that Dicer had sloppily processed their single shRNA design into as many as 95 completely separate, distinct RNA strand variations.
95 different variations from one single hairpin design. That sounds like the quality control inspector completely fell asleep at the wheel. If the cut is that messy, and we know that shifted seed regions cause horrific off-target toxicity like in the rat brains, shouldn't 95 different strands have caused massive, uncontrollable off-target liver failure?
That is the biological paradox that stunned researchers. Despite generating 95 different sequence variations, the TT034 drug was remarkably safe and well tolerated in primates, and later in the phase 1 and phase 2 human trials. It didn't destroy the liver.
How is it physically possible?
It requires looking deeper into the rules of RISC loading. Further thermodynamic analysis showed that while Dicer produced 95 physical variations of the strand, the vast majority of those strands were thermodynamically unviable. They lacked the proper stability profile at their 5' ends.
So they were duds.
Exactly. When AGO2 tested them, it immediately rejected them and shredded them. Very few of the 95 variations were actually viable enough to be loaded into the active RASC complex, and the specific ones that did load successfully were the ones that effectively cleaved the hepatitis C target without causing widespread off-target damage. It challenged the accepted dogma. It showed that nature is inherently messy, but it has built-in thermodynamic buffers to handle its own mess.
So we slow down the factory with weak promoters, and we rely on thermodynamic buffers. Is there anything else bioengineers are doing today to make these drugs safer? I see a note here about shmears.
Yes, shmears represent the current cutting edge of safe design. To further reduce toxicity, modern bioengineers are moving away from injecting raw synthetic shRNA hairpins entirely. Instead, they look at the natural microRNAs that the cell already produces safely, like miR30.
Okay.
They take the protective structural scaffold of miR30, hollow out the middle, and embed their active therapeutic targeting sequence deep inside that natural framework. This creates a shmear.
So it's camouflage.
Exactly. Because it perfectly mimics the exact structural nuances, the loops, and the stems of a natural cellular transcript, it is processed much more efficiently by the Drosha microprocessor complex and exportin 5. It seamlessly merges into the natural flowing traffic of the cell's biochemistry rather than causing a massive toxic traffic jam.
Okay, we have covered an immense amount of ground. To wrap up this massive technical journey, I want to zoom out. I want to locate shRNA in the broader landscape of modern genetic medicine. It is obviously not the only tool out there. I want to know how it stacks up. Let's start with its closest cousin, shRNA, synthetic small interfering RNA. We touched on it briefly, but clinically, how is an shRNA drug fundamentally different from an shRNA drug?
shRNA is the purely synthetic approach. With shRNA, you do not use DNA vectors. You do not use the nucleus. You chemically manufacture the final, perfectly cut, double-stranded RNA product in a pharmaceutical manufacturing plant.
So you skip the factory entirely.
Exactly. You encapsulate those millions of shRNA molecules in a lipid nanoparticle, and you inject it directly into the patient's vein. The major advantage is simplicity. The shRNA enters the cytoplasm, bypasses the nucleus, bypasses Drosha, bypasses Dicer, loads directly into RASC, and goes to work immediately. It requires minimal processing by the host cell.
That sounds significantly easier and safer than messing around with viruses and DNA inside the nucleus. Why not just use shRNA for everything?
Because shRNA is strictly transient. It does not last. Once the synthetic shRNA is injected into the cytoplasm, it immediately begins to degrade due to cellular nucleuses. Furthermore, as the patient's cells naturally grow and divide, the physical concentration of the shRNA drug is diluted in half with every cell division. The clinical pharmacokinetic data shows that cytosolic concentrations of shRNA can plummet 100-fold within just 48 hours.
So it wears off incredibly fast.
Exactly. That means the patient requires constant, recurring, often highly expensive infusions of the drug every few weeks for the rest of their life just to maintain disease suppression. shRNA, on the other hand, because it uses a vector to install a permanent DNA manufacturing plant inside the nucleus, is essentially a one-and-done continuous therapy. The cell makes its own drug forever.
Okay, that makes sense. What about the massive elephant in the genetic engineering room? CRISPR-Cas9. The sledgehammer that won the Nobel Prize. Everyone thinks CRISPR is going to cure everything.
CRISPR-Cas9 is undeniably revolutionary, primarily because it edits the master DNA genome permanently. But, as we discussed briefly with the CAR T-cell multiplexing example, it relies on violence. It works by physically breaking both strands of the DNA double helix. When you break DNA, you rely on the cell's internal emergency response, a process called non-homologous enidae, joining to repair the break.
And you mentioned earlier that's prone to errors.
Highly error-prone. The genomic data shows that nearly 1/3 of the resulting CRISPR repairs fail to achieve a clean, functional knockout of the target gene. And far worse, Cas9 is known to make cuts at off-target genomic locations.
And if CRISPR makes an off-target cut in the DNA, that's it, right? It's permanent.
Yes. It causes permanent, irreversible structural damage to the patient's genome. Once you make an off-target edit with CRISPR in a living patient, you cannot undo it. shRNA never touches the DNA. It only temporarily intercepts the disposable mRNA copies in the cytoplasm.
The CRISPR field knows this, and they've evolved. What about the newer iterations? I'm reading about CRISPR, or CRISPR interference inactivation. These don't cut the DNA, right?
That's correct. CRISPR uses a catalytically dead Cas9 protein. It retains its ability to find a target, but its molecular scissors are broken. It doesn't cut the DNA. It simply parks on the promoter region of a gene and physically blocks transcription from happening. It is significantly safer because it avoids double-strand breaks. However, the limitation of CRISPR is efficacy.
It doesn't work as well.
It often struggles to reduce the target gene expression to absolute zero. It leaves behind residual amounts of the disease-causing protein. Additionally, parking a massive, bulky Cas9 protein complex on the genome can cause steric hindrance. It physically crowds the DNA, which can accidentally interfere with the transcription of neighboring vital genes that happen to be nearby.
And what about Cas-Arcs? This is a CRISPR system I read about that specifically targets RNA instead of DNA. Doesn't that make it a direct competitor to shRNA?
Cas-Rx uses a different enzyme called Cas13. Because it targets RNA in the cytoplasm, it successfully avoids permanently altering the master DNA genome. But it harbors a terrifying biochemical flaw known as collateral cutting.
Collateral cutting, like friendly fire.
Exactly like friendly fire. When the Cas13 enzyme binds to its specific target RNA, its internal cutting domains, the HEPN domains, become hyperactive. Once activated by the target, the enzyme essentially panics and starts indiscriminately slicing any other bystander RNA molecules that happen to be floating nearby in the cytoplasm, regardless of their sequence. It completely loses its specificity.
Oh, wow, that sounds bad.
Because of this collateral damage, Cas-Rx has shown significant in vivo cellular toxicity and cell death in animal models. It is not nearly as clean or precise as the AGO2 cleavage mechanism used by shRNA.
Wow, okay, let me try to summarize this landscape. If shRNA is like renting a gas generator that runs out of fuel after 48 hours, and CRISPR-Cas9 is like taking a massive sledgehammer to your home's main circuit breaker to turn off a single light bulb, shRNA is like hiring a master electrician to come in, open the walls, and permanently install a highly precise, smart dimmer switch on that one specific light.
That is a very highly accurate analogy if we connect this all back to the bigger picture of medicine. For severe diseases that require lifelong, continuous 24/7 suppression of a toxic protein like the slow neurodegeneration of Huntington's disease or the relentless replication of chronic viral infections, but where permanently breaking the host DNA with CRISPR poses far too great a safety risk, shRNA sits in the absolute Goldilocks zone of genetic medicine. It provides continuous, potent, highly specific suppression from a single durable dose, all while leaving the master genetic code completely untouched.
And there we have it. We have traveled from the deepest structural architecture of the nucleus, past the transport bouncers and the thermodynamic quality control inspectors of the cellular factory, to allele-specific snipers, meticulously picking off mutated Huntington's genes without harming the healthy ones, and full silence and replace genomic overhauls for muscular dystrophy. We've seen how bioengineers are tricking advanced tumors by blinding their defenses and aggressively sounding immune alarms, all while carefully navigating the incredibly fine thermodynamic line between a miraculous cellular cure and fatal cellular toxicity.
It is a profound testament to the relentless, iterative, problem-solving nature of biological science. Every single failure, like the early toxicity models that killed the mice, provided the essential data that paved the way for elegant, highly refined solutions like weakened promoters and camouflage smears.
Exactly. The failures were just stepping stones to the actual cure. Now, before we sign off, I want to leave you, the listener, with a final, slightly provocative thought to mull over. Think about your own biology. We've learned today that this entire complex system, RNA interference, dicer, RASC, Argonaut2, evolved in our ancestors over millions of years simply to fight off ancient, invading, double-stranded viruses. Now, we are artificially reprogramming that exact same ancient viral defense system to cure our own internal genetic spelling flaws. But if we have the technology to safely install these permanent dimmer switches into our cells to turn down toxic proteins, what happens when we start using them not just to cure a recognized disease, but to deliberately turn down the genes that dictate the cellular aging process itself? What happens when the microscopic assassin's hit list expands to include the very proteins that cause your cells to grow old and die? You ponder that.
A profound and potentially society-altering question for the future of the field.
Thank you so much for joining us on this incredibly deep dive. We'll catch you on the next one.